Good afternoon and welcome back at ECCMID TV, brought live to you here from Pavilion 1 at the ECCMID Congress Center here in Lisbon, Portugal. It is with great pleasure that I can introduce you to my next guest, who is joining us here in the TV studio via live stream all the way from Cambridge, USA. Professor James Collins, welcome.
Thank you. Thanks for having me.
Thank you. Professor Collins is the Tamir Professor of Medical Engineering and Science and Professor of Biological Engineering at MIT, and has a very strong track record on his research using synthetic biology to create next generation diagnostics and therapeutics, which is why he has also been invited to give a keynote lecture here this year's ECCMID, which will take place, I believe, at 2:45 today in Hall A, so in about an hour and 45 minutes. We're lovely to have you here at ECCMID TV, Professor Collins. Could I interest you to share just a little bit of your keynote lecture without spoiling too much for all our viewers who are actually attending the session? The title is Harnessing Deep Learning and Synthetic Biology to Fight Pathogens. I'm correct?
That's correct. So I'll be covering two general topics. The first is really describing the Antibiotics AI project, which is a recently launched effort we have at MIT, where we're using AI, specifically deep learning, both to discover and design new antibiotics. And I'll walk the audience through the procedure we use, as well as several different case studies, some of which have been published and most of which has not been. In the second part of the talk, I'll actually speak to our efforts in synthetic biology. So synthetic biology is still a relatively new field that's bringing together engineers with molecular biologists to use engineering principles to model, design, and build synthetic gene circuits and other molecular components, and to use these to rewire and reprogram living cells and cell-free systems, endowing them with novel functions for a variety of applications. And I'll share in the talk different efforts we're using to engineer living cells to serve as living diagnostics and living therapeutics, and more recent efforts to use synthetic biology to create paper-based and wearable diagnostics for various challenges in infectious diseases.
So which advantages does synthetic biology have to what we used to do? Why is it possible
I think a key advantage is that we really now provide control. So say what we used to do would be under the banner of genetic engineering, where really the primary interest was to introduce a foreign gene from one species into another and then to produce it at a high level so that you'd have sufficient quantities of a protein for a biotech or clinical application. Synthetic biology now provides control so that you can actually regulate the expression of that gene in response to different challenges, whether it's in response to the presence of a pathogen or in response to density. And so we're now giving really significant levels of control that now are in a programmable fashion that allow you either to design smart therapeutics and or to design highly sensitive diagnostics.
Yeah, so does it have many applications in your opinion? Or
It
can
does,
it?
yes.
Yeah.
Yes, so we have been demonstrating, for example, you can engineer bacteria, so engineer microbes to detect and treat infections, including cholera infections. And just this past week showed that you could engineer bacteria to prevent antibiotic-induced gut dysbiosis and thereby also prevent C. diff infections. On the cell-free side, we've actually demonstrated that you can generate diagnostics for detecting antibiotic-resistant infections, for detecting Ebola, for detecting Zika, for detecting COVID-19. So there are many, many different applications, some of which we've already brought to the market as real products.
So that's very interesting. And you'll also be leading Fireplace session right after the keynote lecture, I believe. Are you looking forward to interacting with maybe investigators who are a little more earlier in their career about this topic on deep learning and synthetic biology?
I am, yeah. I mean, I'm always excited to talk with colleagues, and certainly young colleagues. I think we have a real challenge and a real need to excite young people about this space. So at MIT, the young folks coming in to study life scientists are primarily interested in neuroscience and cancer research, two areas of great import, two areas with great excitement. But I think in part what the pandemic has demonstrated, and I think this conference has also highlighted, is that there are tremendously important, exciting, and tractable problems in infectious diseases for which we need a lot of young talent to come in and bring their talents to bear on these big challenges.
Yeah, yeah, that's interesting. So did you manage to follow bits of the ECCMID scientific program yourself, being all the way across an ocean? I can imagine it's very different from being here or attending here on
Yeah, it's
site,
really
but
quite different. I wish I could be there in person. It's
yeah,
nice that the world is reconnecting and getting back. And I think if it was in just a few more months, I could have been able to swing it. I've caught a little bit, but not as much as I probably should have. But the
yeah, well,
program is
it's
really
wonderful
impressive.
that we have the opportunity to do it like this,
Yes.
and we can actually reach a larger audience by having a hybrid event. So are there any other things you're specifically interested in yourself being presented here at ECCMID outside of your own research topic?
You know, I'm always intrigued on the new technology and new technology platforms, and certainly we've seen an explosion in diagnostic efforts as a result of
Yeah.
the pandemic, and always also intrigued on what new therapeutic modalities are being explored. So it looks to have been already quite exciting, and I'm really excited to see what else is coming down the pike
Yeah.
from the conference.
Really on top of the pipeline.
Yes.
Well, thank you so much for giving just a little insight in well, the keynote lecture you'll be hosting or be giving just later on today. Thank you for coming online here with us in the TV studio to promote it a little bit, and I hope you have a great session and also a great adjacent fireplace session, and wish you a very nice rest of your ECCMID.
Great. Well, thank you very much. Thanks for having me on your show.
Thank you to Professor Collins. We will be tuning out here at ECCMID TV also, and we will be back at 2 o'clock when we will be inviting to our TV studio Professor Eskild Peterson, who is the chair of the Emerging Infections Task Force. So tune in again at 2. See you then.